This screencast is on the collection and analysis of hair evidence found at a crime scene. So let's take a look at the biology of hair. Hair is composed of the protein keratin. Keratin is also the primary component of fingernails and toenails, just to kind of give you a comparison of what we're looking at. Hair is produced from a structure called the hair follicle. Humans develop hair follicles during fetal development, and no new follicles are produced after birth. So all the hair you're going to have is determined before you're born. Hair color is mostly the result of pigments. Pigments are the chemical compounds that are present in the hair structure that reflect certain wavelengths of visible light, which gives you the particular color of hair that you have. Hair shape can be round or oval, and its texture can be curly or straight. And these characteristics are heavily influenced by your genes, okay? You can change the physical appearance of your hair. Some of those changes can be affected by nutritional status, but it can also be affected by your intentional alterations. So heat curling, perm, straightening, coloring your hair can change the shape and the texture as well. The body area, so your head, your arms, legs, back, etc. So the particular area of the body from which a hair comes from, okay, from which it originates, can be determined based on the sample's length, okay, its shape, size, color, and other physical characteristics. We can test DNA from hair. If we're looking for nuclear DNA, then we must have the root present. We can also test for mitochondrial DNA, which would be DNA that's found strictly in the mitochondria inside of the cell, and that can be tested for whether the root is present or not. So here's a sectional look at hair structure. Hair is composed of three principal parts. Okay, the first part here is the cuticle. The cuticle is the outer covering. Okay, it's going to have distinctive patterns which let investigators determine the species that the hair is coming from. It can also help with determining chemical treatments that have been done to the hair, disorders or drug therapy, and age, infant or elderly, of the donor. Next, we have the cortex, which is going to be a protein-rich structure around the medulla that contains the pigment. The cortex is the middle section and the longest or largest section of the hair, and it shields the medulla and contains all the color pigments. Okay, here's the medulla, which is the central core of the hair. It can be present in a variety of patterns and thicknesses that can aid in identification. If you want to think about the hair structure as a pencil, that's kind of a helpful analogy. Think of the structure of the pencil. In the very center, we have a lead core, and that would be like the medulla. And surrounding the lead, we have the wood of the pencil, which would be like the cortex. And then on the outside of the pencil, we have the paint. So for whatever color pencil you're dealing with, or yellow for a regular lead pencil, that would be the cuticle, the outer coating. Okay, so just a little analogy to help you understand the structure of hair a little bit better. Spend a little bit more time here taking a look at the cuticle. Okay, the cuticle can vary in terms of its scales, its thickness, and whether or not it contains pigment. Okay, so the scales we can see here in this diagram. Okay, on a piece of hair. So when we're analyzing that, we want to think how many scales are there per centimeter? Do they overlap a lot? What is their overall shape? How much do they protrude from the surface? So those are questions we want to look at when we're looking at the cuticle of the hair. And again, like we mentioned in the previous slide, just kind of keep in mind that the cuticle can be very helpful in distinguishing hairs between different species. So is this a human hair or a dog hair or a cat hair? But it's not usually very useful in distinguishing between different people, because humans will all have a similar structure to their cuticle. Next, we're going to take a closer look at the cortex. Okay, remember the cortex is like the wooden part of the pencil. It's all the stuff that's here surrounding that central core, okay, of the hair. The cortex can vary in thickness, texture, and color. Looking at the cortex is probably the most helpful piece of information in determining which person a particular hair may have come from. We want to make sure that we examine the cortex under a microscope, because it can also help us reveal the condition and the shape of the root and the tip of the hair. Okay, and then we also have the medulla. The medulla varies greatly from hair to hair in thickness and continuity. okay, and in opacity. So in continuity, what we're looking at here is this very dark structure in the center of the hair. This would be a continuous medulla. In the next picture over, we see that there is actually no medulla in that picture. And then in the final picture here on the left, we see that the medulla is broken up. It's very fragmented. Okay, so those characteristics can be helpful in identifying hair. And we can also look at the opacity, which is how much light is able to pass through the hair, and that can be a helpful characteristic also. Like the cuticle, the medulla can help us distinguish between hairs from different species. So again, is this human hair, dog hair, cat hair, etc. It's not as useful in distinguishing hairs from person to person, but it can give us some good information there as well. Okay, let's take a closer look at patterns that the medulla can take. The medulla can be continuous. Okay, so that's in picture B here, and we see where we have one long continuous piece of medulla that runs the length of the hair. Okay, it can be interrupted or intermittent. Okay, and we see that in diagram C, where the medulla is broken up. Okay, but it's not in even pieces. So we'll have a long piece of medulla followed by some short pieces, maybe another long piece. The medulla can be fragmented or segmented, and that's where it's broken up in more even, smaller pieces, and we see this in diagram D. Okay. The medulla can be solid, which means that this entire gray area, okay, would be filled with medulla, or it can be completely absent, which is what we see in diagram A, where there's just no medulla whatsoever. Okay, so the 5 main patterns that we can find for the medulla are continuous, interrupted or intermittent, fragmented or segmented, solid or none. Additionally, we can analyze hair based on characteristics from different racial groups. Okay, so we see here at the very top of this diagram, okay, so this hair sample at the top, this would be a hair from an African American individual. It's going to have a coarse cuticle. The shape is, the shaft is flat in shape. It's going to be extremely wavy or curly. The hair will appear opaque under the microscope because there are dense clumps of pigment and there's an irregular particle shape. Okay, if we move down to look at this portion of the diagram, the center, this would be an example of a hair from someone of Asian descent. Here the hair shaft has a large diameter and is perfectly round. The shaft is also straight and moderately coarse. It has large pigment particles which occur in patches around the medulla, which we can see in the picture on the right. And it will appear reddish to brown under magnification. And then at the far bottom here, we see a sample of hair from someone of European or Caucasian descent. And here the hair shaft will range from round to slightly oval. The greatest variation in pigment and texture shows up in European or Caucasian hair. It's going to range from fine to moderately coarse, straight to moderately wavy, and small to medium-sized pigment particles in a wide range of color classes. So there are a lot of different characteristics we can learn about an individual based on the hair samples that are left behind. So now it's time for you to examine some hairs. What I'd like you guys to do is take a look at these pictures and do some research and see if you can identify the animal hairs that are shown in those samples. So think about, in which samples are we viewing the cuticle? How do they compare? In which samples are we viewing the medulla? How do they compare? What characteristics can be used to identify hair samples? So what I need you to do is list A through I, do some research and try to figure out which animal the hair came from. Write your answer down on a piece of paper, and then especially answer question number 3, what characteristics can be used to identify hair samples? When you're finished, take a picture of your answers and upload it to Schoology. Once you have submitted your assignment, you will be able to view the answer key to check to see how you did.